This is something that I plan to cover in the last lecture actually. So you will see that in the PDF file I have given Shakti, it's actually included as part of lectures 1 and 2, okay, because this forms the end of the part where I model a single neuron, and the lecture 3 was supposed to be all about networks, all right. So but I got delayed, and so what is this? So all through up till now I have assumed that the neuron is like a point, right, but in reality the cartoon neuron, of course neuron don't really look like this, every neuron is different, as Upi showed in the famous pictures of Ramani Kahal, but in high school this is the kind of thing that we used to learn as neuron. So this is your cell body, okay, these are dendrites, and this is the axon, and then here it forms the synapses with the postsynaptic neuron, and here it forms synapses with the presynaptic neurons, right. So the idea is like you have inputs or signals coming in here, and then the outputs are going like this, right. So that's the classic neuron. In reality, of course, this is a very, very simplified model. So you can have synapses pretty much anywhere, you can even have dendrodendritic synapses, okay. So so when you work with a real organism like C. elegans for example, you will find that things are not as simple as this, okay. So but for the sake of simplicity we will just continue with this thing. Now do you see the problem with using Fujinoguma model to model this? What's the problem? The Fujinoguma model is a single differential equation, right, it talks about a single membrane potential, but whose membrane potential I am talking about, because I can put a electrode here, I can put a electrode here, I can put a electrode here, I can put a electrode here. So which membrane potential am I talking about? Is it all same? Well, can't be, right, it's an extended object. So if it's like, if you stimulate action potential here, or actually rather here you are just summing things up, so you are causing activity and the various dendrites, and there's some kind of a summation happening, and then you have an action potential which is traveling along the axon, and then it initiates vesicle formation at the presynaptic neuron, presynaptic end, and then this chemical neurotransmitters would be emitted, and then this would be going and sitting at the receptors at the dendritic end of the postsynaptic neuron, right. So the V here at some xt is going to be different from the V at some x prime at the same time, right. So it's pretty obvious that a single V is a very bad way of approximating a whole neuron, okay. So in effect what we are doing is what Pranay called a mean field, but it's like saying it's a point particle, okay. So why are the two equivalent? Because we are saying that it's so well mixed, so homogenized, that effectively wherever you stick an electrode in, you are going to see roughly the same V, okay, but that's an assumption, okay. So if you are not interested in treating the neuron as a point particle, you have to do something better than this, okay. And why would you not be interested in seeing the neuron as a point particle? When would you be unhappy with treating the neuron as a point particle? Sorry?
If you want to look at multiple synapses that are taking place together.
Yeah, so what kind of questions would you be asking for that to be valid?
A heterogeneous pool of neurons?
No, it's a single neuron, right. So basically you are looking at a single neuron, and the moment you look at a neural network problem, you want to simplify it, right, because it's difficult for you to treat, let's say you are looking at a network of 10,000 neurons, you may want to simplify things such that you have only one membrane potential describing each neuron, right. But if you are looking at, let us say, a pair of neurons or just a few neurons, then under certain conditions you may be unhappy with a simplified description where you just have a single V for each neuron. So what kind of questions are we talking about?
Time of travel down the axon matters.
Yes, why? Under what sort of conditions would this matter?
If the time scale of traveling down the axon and the time scale of the action potential are similar, then there will be a delay just by traveling.
Absolutely correct. So what kind of actual questions, what kind of actual computations are those where this will start becoming important?
If you wanted, if you want to account for that delay in the postsynaptic neuron.
Yeah, so let me, you are on the right track, delay is important, but the delays can be used for actually computation, okay. Actually UP mentioned this, okay. So suppose, so for example, one famous example is the burn owl, okay. So the burn owl uses something like the principle of radar, right. So it basically homes in on its prey by listening to the sound and echolocating it. Now how do you echolocate? We are very bad at it, right. We have a very coarse level of echolocation. We can just manage to make out left and right, which is why stereo works, but that's about it, right. Burn owls are very good at it.
I've seen this case before, so I'm just.
Yeah.
There is a temporal difference in
Yes.
the sound reaching the left ear versus the right ear.
Correct.
So when the whatever machinery inside of the ears
Yeah.
that produces those action potentials,
Right.
there will be a time difference when the electrical signals reach the higher order centers.
Right. So even more than higher order centers, you might actually want to work at the level of a single neuron. So a single neuron will have some dendrites receiving signals from neurons stimulated in the left ear and neurons stimulated in the right ear, right. So that single neuron would be computing the delay in the signals received from the left neuron, from the left ear neurons and from the right ear neurons, and by looking at the delay it will calculate the difference, essentially depending on where it's coming from, the delays would be different, right. Okay, so this is basically covering this what do you call it, azimuth. What about the altitude? Because the owl is in 3D. How will you cover altitude? Anyone?
I think that location of both left and right ears is little different.
Yeah. So left ear and right ear are actually asymmetric. One is higher, one is lower. Okay. It looks horrible from a point of view of symmetry, but it's functionally very important for it. Okay. And so you can see it's basically doing two computations. There are two kinds of delays. One because of the left and right asymmetry, one because of the up-down asymmetry. Okay. And it can make sense. It doesn't all get mixed up because it's doing this computation with multiple neurons. Okay. If you only had a single neuron which computed this difference, then it won't be able to make out which delay is because of the left right and which delay is because of the up down. But because you have multiple neurons doing this computation, it can disentangle what part of the delay is responsible is because of the left right and what part is responsible for the up down differences. And from this it can work out the azimuth and the altitude. So there is an incredible amount of computation going on at the single neuron level. Okay. This is what UP was kind of implying by saying that when we say that networks, there is also a lot of network computation going on at the level of a single neuron. Okay. It is not just networks of neurons. Yeah.
Even if you have two neurons and like one neuron needs the signal they receive would be the same. As in spatially and like left.
No. See, for example, you have two neurons, okay,
Yeah.
and the delays between the left neuron, because you may not be receiving it from exactly the same sensory neurons in your left ear and right ear, right. So different neurons, sensory neurons in your left ear and right ear would be giving inputs to these two neurons. Okay. So the delay calculations would be slightly different in the two. So you basically you disambiguate by looking at all these computations. Okay.
I'll drop you.
Oh, sure. All right. I'm waving my hand here, as you might imagine. Okay. All right. So, in effect, what I'm saying is, for certain kinds of computation, you actually need to know what is the potential at different parts of a neuron. Okay. So, that means we need a spatially extended model of a neuron. It's no longer useful just to have a single V for the entire neuron. Okay. So, one of the simplest cases where you can describe a spatially extended model of a neuron is how does the signal propagate along this axon, right. So, you can essentially take each part of the axon and treat it as a equivalent electrical circuit that Pranay talked about. Okay. So, it is a so each part of the axon, so I can zoom in on each part. Okay, so maybe I should use a different color. So, let us say you look at this one and you look at this one. Okay. Both are described by the equivalent electrical circuit that Pranay talked about. So, it will have a sodium conductance, a potassium conductance, a leak channel, okay, and these will have this reversal potentials, and then finally a capacitor, right. So, this is described by an equivalent electrical circuit, this is described by another equivalent circuit. Okay. And now you need to couple these electrical circuits, this neighboring electrical circuits, because imagine there is another part of the axon which is just next to it, it will be described by electrical circuit, but the electrical circuits are not isolated, right, because the activity of this would stimulate the activity of this, right, because they are just next to each other. So, just through diffusion you will have activity propagating along the axon. Okay. So, this is what is being shown here. So, this what is this, violet, maroon, I am always, or lilac, what color is this? I sorry, it is not a quiz question, I am genuinely like getting colorblind here. Lilac, okay, whatever, pink, whatever, I mean whatever suits you, right. So, this part is the one which is actually excited at this moment, okay. And so, what you see is basically sodium ions are rushing in, whereas this part is actually refractory. So, here your sodium is completely closed off, and slowly the ion pumps are doing their work, so to bring it back to the resting potential. Okay. So, now you will have the action potentials peak occurring. So, if you look at the V here, basically it will be on the upstroke of the action potential, while this is in the downstroke or maybe in the negative portion of the action potential, all right. Now, what is happening is, because this is all cytoplasm, you will have ionic flow along the cytoplasm, right, but diffusion is of course very slow. Okay. So, diffusion is helped along by the regenerative process triggered by the ion channels. So, this is actually a fundamental difference. So, you can think that once you initiate an action potential on a neuron, just let diffusion do its job, right, let it just diffuse through the axon. Big problem. Why? Because diffusion is famously slow. So, if you have a neuron whose cell body is here, and let us say its axon ends here, it will actually probably take days, weeks, years to actually travel from here to here, if I just depend on diffusion. So, instead of that, I have a regenerative process, something which uses diffusion to give me the initial stimulation, but then as soon as this local region crosses the threshold, the ion channels open up, and this very quick process of activation or depolarization followed by repolarization happens. So, it is a wave which is being constantly regenerated. So, unlike normal waves which because of damping will, as it progresses, will gradually become, its crest will become weaker and weaker and weaker, and eventually it will die out. This is like a wave whose amplitude never decreases, okay, it's being constantly regenerated at every point. In nature, do we see such a wave? I mean, not in brain, I mean in actual world out there. Do you know of any examples of regenerative waves? Well, not regenerative waves, I mean basically non-attenuating waves, waves where the amplitude kind of keeps steady. Sorry? Oh, light, okay, hold on. No. So light is too complicated, folks. You know, in fact, we had this kid here who kind of got very angry at one of our faculty members because the faculty member dared to say light was a wave. He said, "What do you mean light is a wave?" He's a high school kid, by the way. He said, "Light is a particle. My teacher told me so." Right? So you know, you'll get in trouble with that kid. So. Any clues? Okay, anyone heard of this thing called tidal bore? Okay, no. Okay, so in nature there are certain waves which have a solitonic characteristics. Okay, so one very famous example of this is when you have a tidal wave going through a narrow channel. Okay, so there are actually very nice documentaries of this tidal bore. For example, in the Hooghly river, which is basically just the Ganges river, Bengalis like to call it Hooghly. Okay, so when this tidal bore comes, you see that there's this wave which just travels upstream along the river, and it's basically non-attenuating. Okay, so why it's non-attenuating? Very interesting question. So people have written down equations, and it turns out to be a fundamentally non-linear effect. You cannot explain it using linear equations. Okay. So there is this equation called the KdV equation or Korteweg-de Vries equation, which shows how a fundamental nonlinearity causes the non-attenuating characteristic of this wave. So we do see such non-attenuating waves even in nature, not just in our bodies. Okay. In the case of the neuron, this non-attenuation is basically maintained by having the wave being constantly regenerated, okay, by the opening and closing of ion channels successively down the axon. Okay. So every time, you know, a part of it shows an action potential, some of the sodium ions will basically, or some of the ions will basically flow into the next bit through diffusion. Okay. Now this is of course very slow, but the moment it goes into that region and crosses the, and makes that transparent potential cross the threshold, it triggers the ion channels to open and you have the V rising up, and so that is why the wave will never die. Okay, it will keep the same amplitude as it travels down the axon, which is not something you would, and it happens very rapidly actually. Okay, so because all you need to do is just have a few ions go to the neighboring region, immediately zoop, zoop, zoop. So you know, you can think of it as if, you know, like well, is relay race a very good example? Maybe not. Dominoes, falling dominoes. Okay, so imagine, like, you know, you have a whole set of dominoes. Okay, I wish I had thought of. I did once set this up with you remember this Jenga blocks. So you can have a whole set of Jenga blocks here, and all I need to do is trigger one, and you will see that there will be a wave which will basically pass through the whole thing. Okay, so nothing actually physically is moving there. What is moving is the disturbance. Okay, so in a similar way, you can have a very rapid wave which will travel through this axon, and so the way you model this is basically having this equivalent circuits which are essentially coupled to each other. Okay, and so that this being triggered causes this being triggered causes this being triggered. So how will you couple them? Okay, equation wise. So this, what happened now? Okay, looks like battery is very weak. This is done by what is known as the cable equation, which Pranay kind of briefly mentioned in his talks. And surprise, 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 cable equation was known way before Hodgkin and Huxley, way before Ramani Kahal even came up with the neuron doctrine. Okay, is Ramani Kahal's idea of neurons basically dates from 1900s, right? Cable equation is 1850s. Okay, well actually 1855. That's when Lord Kelvin actually gave the cable equation. So it's obviously not given in the context of a neuron. Okay, it was given in the context of telegraphic fibers, telegraphic cables actually. Okay, so in the 19th century, telegraph was your internet, right? So suddenly people had this extremely fast means of communication, and actually if you read stuff from the 1850s, magazine articles and so on, you actually see people talking about the information revolution, about how this is going to completely change the world. Sounds very, very similar to the way people talk about internet today or AI today. Okay, so people have been hyped up about information revolutions for a long time. So so Helmholtz actually was the first to show that the signal velocity in nerve fibers is finite. So he measured it as 27 meter per second. Okay, by the way, this is the same Helmholtz as the Helmholtz coil and stuff like that. Okay, and then Lord Kelvin actually described the propagation of electrical signals in long cables, and this was because you had long distance communication by telegraph, and the first transatlantic cables were being planned. So in 1858 the first transatlantic cable was actually laid, which allowed almost instantaneous communication between England and America. Okay, since I'm a fan of completely irrelevant trivia, that one of the first uses of telegraph was actually warning the American authorities that a murderer was actually traveling on a transatlantic liner. And I forget, is this Robert Crippen or okay, so this is so the Scotland Yard basically came to know that the murderer who had murdered his wife for money had vamoosed along with his mistress on a liner. So normally that's the end of it, right? Because there's no way because the only way the information can actually now travel is to send Scotland Yard detectives on the next transatlantic boat, and which will be pretty much a few days away. So by this time, of course, the first boat will reach New York, and they would have disappeared. But and that's what the criminal depended upon, right? But now you had this telegraph cable. So one of the first cables actually sent was Scotland Yard telling New York authorities, please detain this person, and gave a verbal description. And so much to the shock and horror of our criminal, when he reached New York, the police were waiting for him, okay? And he was brought back to England and hanged, okay? Or hung, whatever. So this very exciting story has, of course, a very boring kind of equation, but boring to some eyes. I mean, to others it may be very exciting. So Lord Kelvin actually gave an equation. So if you love equations, you would realize that this part is basically the second order derivative with respect to space, and this part is the first order derivative with respect to time. So if you ignore this, what equation is this? Sorry? Heat equation or diffusion equation. Okay, so this part is actually some kind of a reaction. So this is basically an example of what is known as a reaction diffusion equation. Okay, so in general, sorry, this is really getting boring. Okay, so basically we now go from ordinary differential equations to what are known as partial differential equations. Why partial? Why are they called partial?
Because more than one variable.
V is the only variable there.
Dependent.
The dependence, exactly. So the dependence is not just on time or not just on space, but there's a part which depends on space and a part which depends on time. Okay, so Other famous examples of partial differential equations are wave equation, then Laplace's equation, and so on. Okay. So now we can think of something like continuum of either oscillators or excitable systems where the neighboring points are actually interacting through diffusion, and within each element there is some interesting reaction going on, like for example your electrical circuits are describing what is the activity at each point in space. So this is basically telling you how V will vary in space. So if you cut out this part, there is no spatial part anymore. So that's basically just going back to your differential equation which was describing the point neuron. But now in addition you have some diffusion which is happening through between neighboring points, and that's what is going to cause one region to excite the next region, which in turn will excite the next region, and so on. So once it drives the V above the membrane potential, this local part will take over and you will get an action potential at this region. Okay. All right. So if you want to derive this, actually there is a very interesting and neat way of deriving it, very simple. So imagine that each part of this axon, I label it with some index. So let us say this part is called n, this part is called n minus 1, and this part is called n plus 1. So they are all neighboring regions, right. So I can ask what is this current which flows from one region to its neighboring region. So this I called, by the way, so this is called G gap because you can also use this to model cells which are coupled by gap junction. So for the axon, of course this is just the axial conductance, it is not the G gap, but the current that is flowing across this between the n and the n plus 1th element is the net current. So the current from the nth to the n minus 1th cell is basically used to find out what is the net current that will pass through from one element of the axon to the next element of the axon. So this is basically simply the corresponding axial conductance and the difference of the membrane potentials of the corresponding region. So this is the membrane potential here would be Vn minus 1, the membrane potential here would be Vn, and the membrane potential here would be Vn plus 1. And since the current is basically proportional to the difference of the potential of this region and this region and this region and this region, you can just basically say okay, the net current that flows through is the current that flows from here to here minus the current that flows from here to here. So that's basically just related to the difference of the corresponding potential differences. Okay, and then I can expand this in terms of a Taylor series. Okay, so if you don't remember Taylor series, don't worry. Essentially it will mean that you can expand it in terms of first order derivative, second order derivative, and we keep only up to the second order derivative, and we will find that the first order derivatives cancel out and we are left with just the second order derivative, so that we get the current that passes through the junction as simply the second order spatial derivative of V. And so this finally gives us the capacitance times the first order derivative of potential with respect to time is some axial conductance times the second order spatial derivative of potential with respect to space, and this is the local part which is the ionic current. Okay. So this is just for your edification. I mean if you don't want to remember it, don't, you can just go and use the FHN chain MATLAB file that I shared and you should be able to simulate it, okay, how this wave actually travels through the synapse. Okay. Anyway, the slides will be there, so if you want to, if you have any more questions about this, please, you can, I am always there, even next week you could ask me more questions about how this is derived. Okay, finally, connecting neurons. So this is basically, we are coming to the material for today. Okay, just half an hour late. Okay, so how do we, so this is all about neurons, right? But the workshop is about network neuroscience, which means we are primarily interested in how do we couple neurons with respect to each other. So how do we connect neurons? So I understand Pranay would be actually talking about this in a little more detail. So I will just give you the bare sketches of this. So the question is, how do we model a synapse? Okay, so we have been mostly dealing with how do you model a single neuron's activity. But a single neuron is actually not very useful for our purposes. We actually want a neuron to trigger activity in another neuron. Okay. So that means we either have to model a synapse or we have to model a gap junction. Okay. These are the two ways in which a synapse triggers activity in another synapse or inhibits another, triggers activity in another neuron or inhibits activity in another neuron. So we know, of course, from our biology that biologically the way it happens is the activity when it comes along the axon of the presynaptic neuron, it causes vesicle formation, which in turn causes certain neurotransmitters to be released, and these neurotransmitters go and bind on these ligand gated channels at the postsynaptic neuron, and then this causes ion channels to open up there, which causes ion flow, and then if it manages to push the membrane potential above the threshold, then it will cause an action potential to be generated at the postsynaptic. Okay. So this is roughly how it works. Okay. Just to make things a little more complicated, there can be two types of synapses, excitatory or inhibitory, and this is because of the different kinds of neurotransmitters. Okay. So the neurotransmitters can not only help open up channels, they can also prevent channels from being opened up. So what does inhibition mean? So like again taking an analogy, excitatory and inhibitory synapses could be thought of as just look into a circle of acquaintances. So there will be some people who are very excitatory, I mean in the sense that if you fall into depression, the other ones like, yeah, let's go for a concert, let's go watch this movie, so they're always pepping you up, right? And there are also acquaintances who are, what will happen? Yeah, well, we are not being prepped up for life, we'll all die, what's the point of life anyway? So even no matter how much you're pepped up, they make you depressed, right? So just like that, just like your acquaintances, you can classify into people who are like optimistic, excitatory ones and very, very pessimistic, depressive ones. You can classify synapses, and actually Dell's hypothesis would say that even neurons can be classified into excitatory ones and inhibitory ones. Okay. Dell's hypothesis is not strictly true. There could be neurons which, depending on context, the same neuron can actually have excitatory synapses and inhibitory synapses, but Dell's hypothesis is a very simplifying assumption which says that at some approximation a neuron would only have excitatory synapses, that is they can only give out excitatory neurotransmitters or they can only give out inhibitory neurotransmitters. Okay. So that means you can classify neurons as excitatory, pepping up ones, who always have a rosy view of life, and inhibitory ones, very, very down in the dumps kind of characters. Okay.
You're assuming that's the ground state.
Sorry?
But then you're assuming that's the ground state.
What do you mean ground state?
The zero state, whatever.
No, I'm not assuming it's so basically it depends upon what neurotransmitters you are sending out from your synapse. So you are activated. So like depressive characters will only depress you if they are active. Normally if they are like inactive, they will just sleep in their rooms, not bothering you, but they feel energized to go out and make you depressed also.
So the electrical description of both excitatory and inhibitory neurons is same.
Absolutely.
The,
All of them will show action potentials, but the result of that action potential on inhibitory neurons would be to go and depress you. All right. If you wake them up, basically you shouldn't wake them up. That's the moral of the story. I mean, no, not in the brain. I mean, in society. In the brain, they play a very important role, because if you don't have inhibitory neurons, you are in a big deal of trouble. Yeah. If all of your brain starts get activated, you are just going to like do this. Okay. Epilepsy. All right. So actually, I wonder whether there is a social utility of depressive persons. Oh, but that's another story. Might be a project actually. No, Shakti will really get mad at me now. Shakti is pretty serious about these projects. Okay, so so am I, by the way. I'm just joking. So how would you model this? So now, in addition to the ionic current, you actually have a synaptic current also. Okay, so the synaptic current tells how a neuron which is connected to you via synapse. So basically, you are at the postsynaptic end of a neuron, right? A neuron is basically giving synapse to you. So that means you are receiving some synaptic current, and this will decide whether, if the presynaptic neuron is activated, you will be activated or you will be depressed. So you need some expression for this. So as Pranay already showed, generally a current can be written as, so this is a very famous law in electricity and magnetism. I mean, this general form, what is it? Ohm's law, right? Current equal to conductance into potential difference. Okay, so normally we think of in terms of resistance. So V equal to IR, or voltage equal to current into resistance. So I am just replacing resistance by its reciprocal, conductance. So that is why I can write it as current equal to conductance into potential difference. So you had reversal potentials, Pranay has already covered that. So here you have a synaptic reversal potential, right. So the synaptic current is equal to the synaptic conductance times the difference of the potential drop minus the synaptic reversal potential. Now, looks simple enough. The problem is, of course, where is the problem? The problem is this is not a constant. Okay. Just as Pranay showed that the sodium conductance and the potassium conductance actually have very complicated potential dependencies. Okay. So G synaptic is also itself quite a complicated function. Okay. Now, what is the synaptic reversal potential? The synaptic reversal potential is what decides whether a synapse is excitatory or inhibitory. So depending upon its value, so if it's like, for example, 0 millivolt, it will behave as an excitatory synapse. If it's set to some large negative value like minus 75 millivolt, it will behave as an inhibitory synapse. So if you want to model a excitatory or inhibitory synapse, all you have to do is change the synaptic reversal potential. So depending on that, the nature of the synapse will change. Yeah.
Why isn't excitatory synapse having a positive reversal?
Yeah, good question. Tell me.
The potential difference will become.
So what is the reversal? So reversal potential has a meaning, right? It reverses. So if you cross that, the current gets reversed, right? So does that tell you? So basically, in inhibitory versus excitatory, you want it to react differently. So in the inhibitory one, you want to make sure that you depress the neuron state such that it's incapable of giving action potential even if it's receiving excitatory stimulus from other neurons, right? Whereas in excitatory synapses, you want it to be capable of giving action potentials even if it's not receiving any signals from other synapse. Okay. So this obviously has to be a very negative quantity to guarantee that even if it's receiving excitations from other neurons, it's depressed enough such that it becomes very difficult for it to rise above the threshold. Okay. So that's why you need different values. So this has to be very strongly negative to ensure that others cannot actually excite it.
So all an inhibitory neuron essentially stays not excited.
Inhibitory neurons' role is to make a neuron less likely to be stimulated.
But it does get stimulated, like in rare cases.
Because you could be receiving lots and lots of excitatory synapses from many other neurons, all of which are activated.
And then you pulse.
So it's basically then it becomes a question of a tussle between how many inhibitory people you know and how many excitatory people you know.
I'm not still in peace with the adverbs, but
Okay.
but all it does is that it just stays there.
It has to be activated. If it's not activated, it's not going to do anything.
So on rare occasions where it gets so many inputs, it gets activated.
No. the inhibitory neuron has to be activated for it to do its job.
Okay.
Okay. So suppose you have action potential in the inhibitory neuron. Okay. So now then it sends out inhibitory neurotransmitters from its synapse. Wherever it's binding in the presynaptic end of the neighboring neurons, those neurons now become far more difficult to be excited.
So, it lowers the potential in
Well, okay, so one way to think of it is that it's basically making it far more difficult to excite. So you can think of it as if it's raising the threshold enormously. Yeah.
So the inhibitory neuron which has the action potential to stop the synapse at the next neuron, for example.
Not stop. Basically, the synapse is working, it's sending out neurotransmitters. The neurotransmitter's job is to go and bind to the postsynaptic neuron, making it less likely that it will be excited by stimulus coming from other neurons.
So it would not conduct a signal. It would not let the signal pass through, right?
Which one?
The inhibitory neuron.
The inhibitory neuron has a
Pulse
action.
being an electronic pulse, like the voltage.
See, the action potential has to go all the way to the axonal end
Yes.
of the inhibitory neuron, because otherwise no neurotransmitters will be released. Okay.
But what if we just remove it?
What if you just remove this?
Yeah. Circuit is not completed, so the pulse won't take the other.
No, but then the postsynaptic neuron can get excited. So without this, the postsynaptic neuron would have been excited. But if you
We're
have
not
this,
connecting them to the previous neuron. We just leave a thing. There's nothing there.
no, hold on. Let's just draw this. Okay. So let me draw. So this is my inhibitory neuron, and this is my postsynaptic neuron. It doesn't matter whether it's excited or inhibitory. Okay. So this is receiving another input from an excitatory neuron. Okay. So this is excitatory, this is inhibitory. Okay. All right. Now, if this wasn't there, this action potential would have triggered an action potential here. In the presence of this, it blocks the neuron from being excited by this excited neuron. Is that clear? Yeah.
So the inhibitory neuron to be activated, it is activated by some other neuron.
Yes. So that could be, for example, that would typically be an excited neuron. Yeah.
So in that case, if I have to model that with an inhibitory neuron, I would call it an excitation rather than inhibition.
So here we have an excitatory neuron whose action potential travels and causes this neuron to show up an action potential, right? Okay. Because this needs to be activated for it to do its job, which is inhibition. Okay. Sometimes in biology, you have inhibition of inhibition. So most of the time, control is done not through excitation, but inhibition of inhibition, which is effectively same as excitation, but it gives you an ability to regulate more finely. Okay. So this is actually a standard trick in biology. Even in gene regulation or intracellular signaling, you will find that most of the time positive stimulation is done not through direct positive stimulation, but through negative of negative. So inhibition of inhibition, okay? Because it gives you 2 points at which you can control it. Okay. Yeah.
Yesterday in Search Talk, there was like multiple different cases of behavior. So there is one oscillatory behavior of neurons also.
Yes.
Even in that case, will an inhibitory neuron inhibit, or is it possible that inhibitory neurons can cause an oscillation of? Like,
No,
if
inhibitory
it's a high enough
inhibitory
inhibition.
neurons can certainly cause oscillation, but I mean, what does this?
So can it cause a type of stimulation for the next neuron if there are multiple
No, so there is a very
inhibitors?
simple way inhibitory neuron can actually cause oscillation
Okay.
by doing exactly its job, which is like this. Imagine a population where you have lots of excitatory neurons, lots of inhibitory neurons, and they are all cross coupled with each other and also coupled with each other, right. So initially, let's say there's some stimulation which causes a few excitatory neurons to be stimulated, okay. So what this will cause is that, you know, these guys come together, hey, new Tom Cruise movie, let's all go out, okay. Now this in turn will wake up the inhibitory neurons who come out, another Tom Cruise movie, one more Mission Impossible, blah, and basically they would cause de-excitation, right. So you will see a peak in excitation followed, and this is causing the inhibitory neurons to all wake up, and they are causing the excitation to gradually fall off, okay. Because the excitation is falling off, now the inhibitory neurons also kind of go to sleep again, go back to their room, and the few excited neurons which have been still triggered now, hey guys, let's go, let's go, let's go, excitation rising up, all this shouting is waking up the inhibitory neurons which are again causing, don't tell me you're going to watch Tom Cruise again, blah, and so you will have this up and down, right. So excitatory and inhibitory neurons in a population can actually cause an overall oscillatory behavior at the population level.
But can multiple inhibitory neurons cause a direct downstream neuron to have oscillatory behavior? If there's if they push the voltage of that neuron very far down,
yes,
can that cause an oscillatory?
Yes. So this is called post-inhibitory rebound. I didn't get into it because Pranay will get into it. Okay, but you can actually see it in Fibonacci also.
Okay.
So if you have an inhibitory neuron inhibiting an excitatory neuron, and then at some point the inhibitory neuron's stimulation is cut off, what will happen is all this time it was keeping down the excitatory neuron, suddenly it's released. Okay, so what happens when kids who have been told by their parents never, never, never ever drink fast food, go to a friend's party? They just, completely OD on Coke and whatever, Pepsi and chips, right? So too much restraint may cause a overreaction, right? All right. Okay. Any questions? All right. So how do you do a Fibonacci model with the synapse? So Pranay would be doing Hodgkin-Huxley model with the synapse. Actually, you can even do a Fibonacci model with the synapse. So essentially now you have to model the the synaptic conductance as a dynamical variable. So just as you had M, H, N in the case of the Hodgkin-Huxley neuron, for the synapse also you have a gating variable S, and that gating variable S has a equation very similar to what Pranay was showing for M, H and N. So it has a channel opening rate alpha, channel closing rate beta, and this is the number of closed channels, there is a number of open channels. Okay. So the God is in the details, and so as always it's the expression of the alphas and the betas which depend on the presynaptic potential, which is actually the complicated part. Okay. So you can just look up my slide. So basically it's typically modeled as a sigmoidal or a step function. Okay, I'm not going into the details because the implementation you can find in again a MATLAB code which I've given along with my slides. But again, if you have any questions, please feel free to talk to me later. Okay. Now you can also have gap junctions. Synapses are not the only way in which neurons can communicate with each other. So these are specialized proteins, typically connexin proteins, which come together to form some kind of a gate. Okay, so these gates allow the cytoplasm of two neurons to directly connect with each other. Okay, so this means that directly through diffusion you can have ions transport from one neuron to the other and thereby cause excitation. And so this has this great feature that there is no synaptic delay here. Okay, so
behave like a single neuron.
So it forms what is known as a continuum. Yeah. So very good. This is basically gives you the guess of how we model it. We model it as a diffusion equation. Okay. All right. Okay. In 10 minutes I have to cover. Yeah.
In the case of the electrical synapse, what if the two neurons are of a different type? Like if you had an excitatory and an inhibitory.
Very good question. So because there are no neurotransmitters, guess what?
All of them are excited.
I mean, basically, if one neuron is activated, it will basically cause the next neuron to be activated, right? It's a very good question, and one of my PhD students once did this wonderful experiment where she did, like, what if you have excitatory and inhibitory neurons, which are synaptically connected, but which also have gap junctions with each other, and gap junctions are relatively fast communicators, and you have synaptic delays in the synapses, and so what kind of dynamics will they come up to if you have both gap junctions and chemical synapses working on this thing. So by using different architectures, you can basically see what kind of crazy weird dynamics this will result in. So at a very fast scale, you have people kind of activating each other, and at a very slow scale, they're inhibiting each other. Okay, so they can get up to all kinds of very weird mischief. Okay. All right, so from the really interesting part in 10 minutes. Okay. Okay, so you can, as Upi said, many scales in which you can see neurons and neural networks. So so far we've been looking at not even this, actually more at the cellular level, and I have not even covered the spike train statistics, so I'm really going slow. Then at the complete macroscopic, okay, at the mesoscopic level, you can have a group of neurons talking to each other, so you can do a mean field model. Then you can divide the entire population into excitatory and inhibitory, and show how interaction between them leads to oscillation. Then you can couple them with each other, and then you can have layers. So this is what, for example, Sumitra was talking about, you can have layers of neurons, but of course in her case it was a very simplified connection where each layer was giving connections to the next layer, and there are no connections within the layers, nor are the connections jumping across the layers. Okay, but in general you can have all kinds of connections like this. Okay, and this is very similar to the feed-forward network that Anuran was talking about. At an even higher scale, you can have for example many such groups talking to each other. So this multiple columns, you could have field models where you basically say that you are looking at such a bird's eye view scale that the individual neurons are no longer visible, and you just see it as like a field, a continuum where activity basically spreads as a wave from one point to another. And then at even higher scale, you can look at the whole brain. Okay, so it's now the brain regions are the ones which are the vertices, and the tracts connecting them are basically the connections. So Upi was talking about 2 networks. I think if you include this, you can talk about 3 networks. So the third network being even larger than at the neuron scale, where you are looking at the macroconnectome, where the nodes are not neurons, the nodes are actually brain regions, and the connections are the tracts of like zillions of synapses which go from one brain region to another brain region. Oh, and this is a nice book if you want to get into details of this dynamics. It's not my favorite book, but it's not bad. Okay, so when you look at when you're trying to model brain activity or activity over large areas, typically there are 2 kinds of modeling paradigms you can take. You can take neural mass models. So neural mass models where you are basically looking at a small point or small region in the brain, and you are trying to model it as basically a set of coupled differential equations. So you are viewing that brain region as a point. Okay, again you are basically doing mean field. Okay, you are basically saying it's all homogeneous, the properties actually don't vary from one point within that region to another point within that region, and you just write it down as a set of differential equations. Or you can do a neural field model, which is basically you think of it as a continuum, and you say that okay, things vary in space but in a continuous manner, so that I can write it as some del square v del x square equal to some complicated nonlinear function plus del v del t. Okay, so how do you do neural mass models? So this is a very simple idea of how you can do neural mass model, where you divide one brain region into 2 parts, one consisting entirely of excitatory neurons, another consisting entirely of inhibitory neurons. So let's say all of these use AMP or NMDA neurotransmitter, and all of these use, let's say, GABA transmitters. Okay, so these connections between the excitatory neurons you can say is like consisting of a self connection from this population back to itself. Okay, so you are basically coarse graining all of these internal connections as just one self connection from the population to itself. And similarly, all the connections within the inhibitory neurons you can treat as a self connection from the inhibitory population to itself. And the connections from the excitatory to the inhibitory could be treated as a cross connection from the excitatory population to the inhibitory population, and similarly all connections from the inhibitory neurons to excitatory neurons can be coarse grained into a cross connection from the inhibitory population to the excitatory neuron. And so now what you do is you basically just work with two very simple variables, u and v. U is basically the fraction of firing excitatory neurons in the population, and V is the fraction of firing inhibitory neurons in the population. Okay. Okay. So now that we have this, how do we write down the equation? Okay. So first imagine that this population was in isolation. It's receiving no inputs. Okay. How will it behave? Sorry? It will
continuously keep exciting itself.
I said it's in isolation, that means no self-feedback either.
Oh.
So basically think of all the neurons as isolated. So initially you have made u fraction of them excited. So what will happen? They will just die out, right? And it will be an exponential dying out because you may have timed, stimulated them at different points of time. So you expect that du dt will go as some minus u, proportional to minus u, but what will be the decay constant? There will be a decay constant which is proportional to the refractory period, right? Okay. So this is that part. Tau du dt equal to minus u. So when all the neurons are isolated, this is essentially how it's going to behave. All activity is going to die out exponentially, and similarly for the inhibitory neuron. Okay. So that part is okay. Any questions? No. Okay. Now comes the interesting part. The 1 minus u is the fraction which is not yet excited. So they are free to be excited. Okay. What fraction of them would be excited if you now allow all these connections? That is the question you are asking. Okay. So this function is basically telling you that if you have u fraction of firing excitatory neurons and v is the fraction of firing inhibitory neurons, and you have some external current which is being given to the excited population. By the way, this does not mean that some neuroscientist is going and punching them with a electrode. This could be some excitation coming from other regions of the brain. Anything which is from outside this population would be external current, right? So if you have this, so you are basically trying to write it down as some function of u, some function of v, and this external current. Okay. In addition, you have this connection weights which tells you what is the density of connections of the excitatory neurons to themselves and what is the density of inhibitory neurons to excitatory neurons. Now you will notice I have already made an assumption. The assumption here is that all of this is additive, right? Need not be. Okay. Just the simplest assumption would be it's some linear combination of the fraction of firing excitatory neurons, fraction of inhibitory neurons, and external current. Okay. And then it's just a question of what kind of functional form will it have. So I say, okay, let me see, when this is basically the effective excitation that this population is receiving. So any individual neuron in this population would have more likely to be excited the bigger this is. Okay. But let us say, like when it is very low, it's not very likely to be excited. At some intermediate values there will be almost a linear dependence, and then at very high values it will have to saturate because why? Because the maximal fraction you can excite is 1 minus u. So at max this can only be 1. So its minimum value is 0, max value is 1. So it has to be bounded at both 0 and 1. So the possibilities are it could be a hyperbolic function or it could be a sigmoidal function. Okay. So usually people take it as a sigmoidal function. Okay. All right. So most models of these kinds of things actually take this picture and then do epsilon delta variations on them. For example, the Wilson-Cohen model of 1972, one of the first neural mass models, actually do something very similar but in a different kind of way, and they come up with an equation which looks vaguely similar to what we did. And if you look at the Wilson-Cohen model, it actually over a range of external currents given to the excited neurons, its phase space would look like this. So remember, there are just two equations, so I can have a two-dimensional phase space just as I had a two-dimensional phase space for the Fujinogama model. But now remember, this is not a single neuron. I am talking about a population of excited neurons and inhibitory neurons. Okay. So this is a what you call a coarse-grained mean field model. Okay. So now this is my what you call null-Klein for the DUDT equation. This is the null-Klein for the dVdt equation. Notice that this is no longer linear because both have sigmoids. So this is a sigmoid, this is a sigmoid, and they have an unstable fixed point for some range of external currents, and you would get an oscillation. So depending upon your what is your magnitude of your current, you can get different kinds of oscillation. So as you change your input current, you will see that it goes from being completely lying at 0 to showing oscillatory activity over this range, and then basically showing a very high equilibrium value. Okay. Now it looks a bit dull, I admit, but now if you start doing this, if you take many of these Wilson-Cohen models and connecting them with each other. So here the assumption is each part of your brain can be modeled as a Wilson-Cohen unit. And so what we did was we took the macaque network. So this network is basically the network of brain regions of macaque. Okay. So there is a database on the net called COCOMAC, which basically collects together decades of experimental data on which region of macaque brain connects to which regions of the macaque brain. So by integrating all of this information, you can actually get a database which will give you the macro connectome of the macaque brain. That is, the nodes are the anatomically identified brain regions of the macaque, and the connections are basically the tracts which people have traced anatomically. So typically what they do is they use a staining technique to identify that this part of the brain region has a tract of this thickness going to this part of the brain region. Okay. So basically we take that network and we simulate Wilson-Cohen oscillators on each of these nodes, and as we gradually change the weight with which the different Wilson-Cohen units are coupled, we see that it goes from essentially each of them executing same oscillation but with different phases with respect to each other, to something which becomes much more complicated. See, all that has changed is I've changed my coupling connection, coupling strength, but now instead of becoming more synchronized, they become less synchronized. So now they are showing different frequencies, and then all hell breaks loose when you have even higher connectivity. So if at this high connectivity you basically record the activity of a single Wilson-Cohen unit, it looks like this, which is actually pretty similar to what you would see if you stuck an electrode at any point in a macaque's brain and record a local field potential. The black dots are the instantaneous position of each Wilson-Cohen unit. This is U and this is V. So this is instantaneous snapshot. So this is basically to show that they are all out of phase. There's no synchrony whatsoever. So as the weight, as the coupling strength increases, you actually get more and more desynchronized. So this actually relates to the question you asked Aradhana, right? Aradhana said that the functional network, you asked this question, right? Yeah, someone asked this question that, sorry? Ronnie asked the question, okay, sorry. That how is it that the functional brain network shows higher synchronization if something is weakened? Okay, it can be explained by this model. Actually, as you get lower and lower coupling, you are more and more likely to get synchronized. So it's completely consistent. Okay, and in general, okay, and neural field models. So So how do you do neural field models? Basically you use reaction diffusion equation. Okay, you have this diffusion part and you have the reaction part, and then you have the del V del T part. So someone here mentioned that they are working on CGLE, complex Ginzburg-Landau. Yeah, right. So actually if you take each of these units, these are oscillators, right, and so you can basically write it down as a complex Ginzburg-Landau. So if you look at these kinds of brain models, it will show this interesting patterns, okay, which you can try to match with what you get in fMRI or MEG and so on. Okay. All right, and okay, I am now really running close to matching your patience. I am so sorry, Sumitra. Okay, so now you might say, okay, fine, interesting. Can I do something even simpler? So suppose you are trying to capture some of these EEG activities. Well, just for fun, I am showing you a model which, if you are if you have lived through COVID and was conscious, you may recognize as the infamous or famous SIR model. Okay. All I have done is I have replaced susceptible populations with quiescent neurons, infected population with excited, and removed or recovered population with refractory. Okay. So this is the so called SIRS model, where people go from susceptible to infected to recovered back to again susceptible as they lose immunity. Okay. So here with some rate I am going into excited beta, another rate gamma, which is inversely related to the refractory period, I am going to the refractory state. Sorry, this is the duration of the action potential duration, not the refractory period. And then with the refractory period, alpha 1 over the refractory period, with that rate I am going to the quiescent period. So exactly as I do SIR model, I can write down equations for dq dt, dE dt and dr dt. But wait, q plus e plus r is constant because the total number of neurons is conserved. So I actually have only 2 independent equations. Okay. So I can then just work with this 2 coupled ODE system. I can do the analysis with null clients, and then I would find that the condition for this network to show persistent activity is the product of the action potential duration, the reciprocal of the sorry, what was beta, I forgot. Let me just go back. Oh yeah, the probability of a neuron being excited and the average number of connections that a neuron has, this product has to be greater than 1. Okay. So this is exactly the same as the basic reproduction number that you have in epidemics. Okay. So you can see that you can do a very simple mean field model which resembles kind of epidemic. Pranay, you are looking daggers at me. Am I saying something very bad? This is just for fun, okay, not to be taken seriously, okay, but you can do fun things, okay. All right, so I'm like so out of time. I'm just wondering whether so like just to tell you like what I really wanted to say if I had some time, that once you have this, you realize that hold on a bit, if I can simplify it to just 3 states, quiescent, excitatory and recovered, why not just 2 states? And this is where the binary state neuron idea comes in, you might say, although chronologically this is predates this whole idea of this neural mass models and so on. Okay. So the quiescent is the resting state of 0 and the firing potential is 1, but of course you might say what does this got to have to do with neurons? Well, you can just say that 0 is actually -1, the spin down, and firing is the +1, and then basically you have the the equivalent of the McCulloch-Pitts neuron. Okay, so Anuran already did this, like you had this famous model of easing where he showed that where they showed that if you have no interactions, basically the spins are all arbitrarily aligned, but the moment you have interaction, then in absence of any noise they are all ordered, but the moment you introduce fluctuations in one dimensional array, this will introduce disorder. So there are very interesting arguments why you will have disorder when you have any finite temperature. But the interesting point is this: Ising, who will forever be known for this model, is one of the luckiest stiffs who was ever born. The idea was given by his advisor Lenz, okay? But in most books you won't find the mention of Lenz, it's always called the Ising model. Technically it should always be called the Lenz-Ising model because the idea was given by Lenz. He got this right, but the conclusion he drew was completely wrong. He said, okay, it doesn't show any order in, he forgot to say in 1D, and so therefore this is a useless model, and he left physics to become a high school teacher. I wish I had that luck, you know. I've been working like my ass off and I still don't have a model in my name which will be remembered forever. And this guy got an idea from his guide, took the wrong conclusion and left physics, and he'll be known forever. How unfair life can be, right? So essentially, so this is the model which Anuran already showed. So this is the interaction part, where neurons basically affect each other, whether they'll be up or down, and this is the environment part. So this is basically what is coming from outside the particular population. And Ising was corrected later when Onsager did this famous calculation. Actually, a lot before Onsager, already others had guessed that you would get order when you go beyond one dimension to two dimension. So Onsager showed it rigorously that if you have a two dimensional model where spins are not only coupled to the left and right neighbors but also to the up and down neighbors, then provided your temperature is low enough, you can actually order the whole thing such that below T, below some critical temperature, everything will get ordered. And this is what leads to this very famous order disorder diagram, where as you gradually increase temperature or noise, you go from order to disorder, and the field basically breaks the symmetry. So here when you get order, you can either be all up or all down, both are likely. So any small field can now break the symmetry as to whether this is preferred or this is preferred. Okay. Now this brings us to this question of transition. Okay. So you can have two kinds of transition. One is basically, if you just change the field slightly, you can go from all down to all up. So this is a discontinuous transition, right? Suddenly you have a complete abrupt transition. So this is a bit like what we call first order transitions in physics. So these are typically induced by the field. Okay. And to give an analogy, go to your Chennai Central Station. Okay. Typically you will find all these people in this hall kind of doing their own things. Then suddenly, and typically people staring up at the screen and so on, you get this advertisements for RMVK saris and jewelry and all these kinds of things, and then suddenly train number 12110 Mysore local platform number 6 mein aa rahe hai, and suddenly you find large scale streaming. Suddenly order congeals out of disorder. Okay. It's completely discontinuous. Okay. By contrast, you can have a self-organized transition where you have a cooperative phenomena where there is a continuous transition from order to disorder. And an example of this is shown here. So if you want example of this, just during lunchtime, I suggest you do this very, very rigorous experiment. After lunch, come out of the canteen, the more people do this together, the better. Stare very hard at the top branch of one of the trees. Very soon you will find some other people coming and staring with you. Since they don't know you, probably they won't ask you what's up, but anyway they'll look at it. And once a sufficient crowd has gathered, quietly get away. Okay. That's a self-organized transition. Right? There's nothing going on top of the tree. There's no field. Okay. but the very fact that people get curious about what other people are looking at is enough to induce an ordering, right. Okay, so this basically was to tell you a little bit about how all of this actually goes to in the Hopfield model, which Anuran has discussed in detail. So Hopfield model actually has very interesting transitions, which shows how we can settle down to this energy minima, which corresponds to the memory patterns, and you can also introduce noise here. So this noise is basically the stochasticity in neuronal firing. So neurons don't always fire only if the excitation threshold is only if they receive enough input. Sometimes they just accidentally cross the threshold, and sometimes when they receive enough input they may not cross the threshold. So this is basically noise, and you can study what is going on. So this was basically to show how we do noise simulations by the famous Monte Carlo method, which was actually first thought of by Stanislaw Ulam while he was recovering from a surgery. And the name Monte Carlo method is because of Nicholas Metropolis, because it's like gambling. So this is a like a very quick intro to Monte Carlo, where basically the question you're asking is, do I flip or don't I flip? Okay. So you basically look at a spin and you ask, is the energy change greater or less? If it is if the energy change causes to go to a lower energy state, you accept that move. If it is greater, then you accept it with some probability, which is proportional to the energy difference and the temperature. So in this way you can go from, as you gradually increase the temperature, you can go from something which is very well ordered to something which is completely fragmented. Okay. Okay, I really should stop now, but this is where I wanted to start Markov, but I'm so sorry, Sumitra. Okay. Basically I was planning to show that now that you have noise, you can actually now forget about all the deterministic part and just model it as going from one state to another with probabilities. Okay. Okay, so let me stop at this point and